Good morning. I'm going to talk about our work today on understanding aflatoxin contamination. And what I want to talk about is listed here. I want to give an introduction, rationale, and some of the background experiments that led us down this path. Talk about the field experiments that we did this last summer, and give you some results on spore trapping that we've done, and also diverge a little bit from what I'm supposed to talk about and report on some work I did for Syngenta involving two hybrids for reducing aflatoxin contamination, and then finish up with the future work. Well, this is where I work. My lab's here on the 4th floor, office is down the hall at LSU in Baton Rouge. If you ever come through and can find a parking place, come up and see me, and we'll show you what we're doing there. And the rationale for our work comes from this series of ideas. We showed some time ago that touching or physical interaction is necessary for intraspecific aflatoxin inhibition. We also showed that there's specificity in this touch inhibition. Just because non-toxigenic strain inhibits toxin strain A doesn't mean that it can also inhibit toxin strain B. And this work was published in PLOS 1 in 2011. The upshot of that is that each non-toxigenic biocontrol strain has a spectrum of toxigenic strains it can and cannot inhibit. In order to be able to protect corn from contamination, a mixture of non-toxigenic strains which together have the ability to complement each other's lack of inhibitory ability appears to be the best approach. There also appears to be specificity in which toxigenic strains in the soil can infect or not infect corn. Not all of them are equal. Some can, some can't. And if we are able to know the infectious, aggressive strains in the soil, those which can infect kernels or characteristics of those strains, that'll give us a target against which to select for the most effective biocontrol strains. And in order to determine the infectious aggressive strains, we are spore trapping to determine which strains fly, we call it, disseminated on the wind, and how and when our biocontrol strains are flying. Okay? That's our rationale. Some of the early work that supports this came from work we did. We had a collection of non-toxigenic kernel isolates that we screened against a single toxigenic kernel isolate in this system. This is a glass fiber disc, put 9,000 canidia on here. Each of these 50 individually against this one. Let them go 5 days in the dark, take them out, extract the toxin. And what we found is 12 of those 50 completely inhibited toxin production by 53. That in itself is some indication of specificity. Not all of them could inhibit 53. We went on to look at this in a different way, and we took those two strains, 51 and 53, and looked at them in a filter insert plate well system. 51 is the non-toxin producer, 53 is the toxin producer. Put the toxin producer in here, the non-toxin producer outside that. That's a plastic cylinder with a filter in the bottom. 0.4 or 3 micron filter. There are two compartments there that physically separate those two strains. Nutrients can go back and forth, any signal molecules can go back and forth. When we did that, this pair, which was completely inhibitory in the suspended disc, now made toxin. So we took them, mixed them together, put half of them in the insert, half of them in the well, and at the end we established no toxin like we had in suspended disc. This told us that in order to shut down toxin production, these organisms need to interact and touch in a physical way. And we also showed that there's specificity in that touch. Well, what does this mean for biocontrol? This is some old data, and this gets to the point of using multiple strains of atoxigenic isolates. This looks at treatment of a field in St. Joe with either Aflatoxin or 17, 19, or 21 isolate, which were one of those 12 that I mentioned earlier. Each of those alone didn't do much to inhibit aflatoxin in this experiment. But when we mixed 17 in Aflatoxin, 19 in Aflatoxin, 21 in Aflatoxin, all of those were much superior to any of them alone. And the purpose of our work here is to try to demonstrate that on a large level in multiple states. And I want to talk about our barley inoculum biocontrol experiments we did last year in Louisiana and Mississippi and tried to do in Arkansas, and a spray inoculum biocontrol experiment that I did in Louisiana. Okay, now the bad news. This is what we found in Louisiana last year. 3 parts per billion aflatoxin, and that was only on one strain. Nothing worked. If you worked with aflatoxin before and inoculations, and you know it's highly variable. This is not very useful data. It doesn't support any hypothesis. This is Tom Allen's data from Mississippi up in Stoneville. It doesn't look much different than ours. He got a 3 parts per billion too, but it's a different combination. And he didn't get much in the way of contamination. Overall, both of those experiments were highly disappointing. The Bloomfield test in Arkansas was flooded out, no data is available. Bert said he'd give me a slide if I wanted it, but I didn't think I needed to show you. Conclusion, Louisiana and Mississippi show negligible contamination. The environmental conditions were not conducive to aflatoxin production.
Did y'all not put out any inoculation or anything?
David, we put out a Tox 4 strain, which is our predominant toxigenic strain that we found in previous years, and as I'll show you next, when we sprayed it on, we did a similar experiment at Ben-Hur in Baton Rouge, our research station. The previous work had been done on treated barley, put between the rows, 10 pounds per acre. This was spraying Canidia directly on silks, coming back a week later and spraying our Tox 4 strain on. When we did that, Tox 4 gave us a 20 part per billion contamination. There's a lot of variability in this. Tox 4 plus 19 was very effective. Tox 4 plus 51 was fairly effective. Tox 4 and Aflatoxin wasn't any different than Tox 4 alone. This one really went up. Tox 4 and and 17 and Aflatoxin. The combination didn't work. Here the combination was a little better. Here the combination wasn't very good. So only had 3 treatments there which really showed any efficacy in that result. So I want to move on now to spore trapping. Remember we said we're interested in what flies. In order for biocontrol to work, it's got to get to the silk, infect, and be there to protect. So we figure one of the things that we need to look at is what flies and when it flies. This is a conidial head of A. flavus. They make these chains of conidia. The oldest one out here, the youngest one down here. these dehiss and picked up by the wind, blow to the silks, and we trap these using what's called the ionic spore trap. This was a device that a colleague of mine developed. Basically, it's run by a 12-volt battery with a timer here. The air, it's hung about ear level in the field. This is the field where we did those experiments at Ben-Hur. It has a fan in it which the first 5 minutes of every hour for 16 hours a day, from 5 in the morning till 9 at night, it takes a sample. Sucks air in under this dome. There's an open cylinder down there. In that cylinder is a wire brush that kind of looks like a gun cleaning brush. Then it has a charge on it. And below that is a stub from an SEM that's used in scanning electron microscopy that has the opposite charge on it. So any particulate blowing through there acquires a charge and it's drawn to the surface of that stub. And that's the basis of this spore trapping. And what I want to try to show you here is that little metal stub that was in there that accumulates particles that are going through that trap for 5 minutes every hour, 16 hours a day. We put that in a sterile tube. We take it to the lab, put in a little SDS, vortex it to remove the attached conidia, and we plate that liquid suspension on AFPA selected medium plate. Count the colonies. Over about the 30 days we ran this experiment, we got over 1,000 colonies. 10 per plate. We randomly picked and isolated, single spored, gave us around 300 isolates. From those isolates, we extracted DNA, quantified it, did an SSR PCR. This is simple sequence repeats. These are repeats in the genome that when the DNA replicates, sometimes there's slippage and you get different size amplicons for different strains. And we use that to try to identify what strains we had trapped. To do that, we ran them on a gel, analyzed them, determined the putative identity. And this was done again in the Ben-Hur field where we knew the identity of the applied isolates. And this is just an example of one SSR. This is strain 4, 8, question mark, and 51. 4 is our toxigenic strain, 8 and question mark were 2 strains that we found in the soil only, never found in kernels, and 51 is one of our biocontrol strains. So each of those has 1 or 2 or 3 bands, and we putatively identify what's trapped on that stub by using this approach, and we sometimes use as many as 3 SSRs per isolate. This is an Excel spreadsheet showing the time, wind direction, number of colonies, and these 10 entries over here represent so far what we know about what we trapped. And to make that a little more legible, out of those 295, we've characterized 186 so far. 26 out of 295, or around 8%, are Aflatoxin. VCG4, which is our toxin producer, is pretty highly represented. 132 out of 290, 45% of what we trapped is the toxigenic strain. 8 and question mark here were ones, as I said, were only found in the soil. We didn't find them in kernels. They didn't fly very well. We didn't find any of this one. Only about 2% of 5 of 295 were VCG question mark. We still have to do 17 and 19. We've done 51, 7.5%. The point here is 7-8% of these 2 biocontrol strains is what we captured flying in the field. We have a great preponderance of the captured spores being the toxigenic strain. So eventually what we want to do with that is determine when they fly, look again at the frequency of flying. It's conceivable that we could select for biocontrol strains. If we had strains which we knew were inhibitory of aflatoxin synthesis in vitro, we could put these on to wheat, say take 10 of them, put them onto wheat, throw them in the field, put the trap out there, let the trap catch spores and ask which one flies best. We got to have more than just flight though, we've got to have ability to infect, but that would let us know which were the spores that became airborne and therefore had a chance of infecting and protecting in the for biocontrol. Now I'm going to switch again, talk about hybrids for reducing contamination. This is a different form of biocontrol. I did an experiment for Syngenta where we used two hybrids. This is a more or less isogenic lines. The difference is this one, Viptera 311, contains their insect control package, 2 cry genes and a VIP3A gene. The difference is the cry genes are made in bacillus when it makes spores. The VIP3A is a vegetatively produced Bt toxin that is made throughout the life of the bacteria, but those 3 genes are in this. And when we did this experiment up at the Northeast Research Station in St. Joe, basically that experiment involved mirror image treatments, these 6 and these 6, and what we were testing was the effect of various insecticides and fungicides, which aren't important to what I want to show you. The 2 high lines here are 3000 GT, 1 in 7, highly contaminated with aflatoxin. These 5 and these 5 are, this half is treated with Aflagard, this half wasn't. But the difference really is these 10 plots times 4 all had, with the exception of this one, low aflatoxin. And this is the dogma that you control insects, you control aflatoxin by limiting access of the fungus to get in through insect wounds. This is the fumonisin results of those 2 hybrids. Again, 3000 GT without the insect control package. Here are the 8 or 10 treatments with the package, and again, quite a difference there in fumonisin levels. If you go to the Syngenta website, they show you the same data. 3000 GT versus Viptera. Well, I talked to my colleagues up at Northeast. They said it wasn't a very bad insect year up there. It didn't have much pressure. And that got me to wondering, this is all conjecture at this point. Excuse me. My thought was, has anybody ever looked at the effect of cry proteins or VIP3A protein on the fungus itself directly? There's a lot of data that says for fumonisin this is true. The Bt suppresses fumonisin. That's the same dogma for aflatoxin. I've emailed the people at Syngenta a couple of times. They said they're going to get me the information. But I'd like to get my hands on some of their E. coli constructs that make those 3 proteins, put them with A. flavus or Fusarium verticillioides and see if there are any direct effects on the fungus or on its ability to make toxin. So that brings me to future work. what we want to do is develop a high throughput industrial strength assay for aflatoxin production, which can be used to screen for mutants affected in ability to touch inhibit or to be touch inhibited. And hand in hand with that, if we're going to get mutants, we need to develop a good mutagenesis system. And we hope to develop an agrobacterium insertional mutagenesis and selection system for Aspergillus to be used to generate mutants impaired in touch inhibition. And we've been greatly aided in this part of it by Jonathan Smith, who's at the University of Arkansas in Bert Bloom's lab. He's made a couple of plasmids for us that we have in agrobacterium, and what agrobacterium does, it randomly inserts DNA into, in this case, Aspergillus. It'll insert in the genome and it'll carry a selectable marker, an antibiotic resistance gene. We have a gene for bleomycin resistance and the phosphenothrycin gene that allows you to select for, say, liberty resistance. So we hope to use those, and what we've done so far, again, we owe to John Smith and Arkansas's work. It turns out Aspergillus likes pina coladas. This is Coco Lopez cream of coconut, and apparently Jonathan and Bert are using this medium to grow A. flavus, look at fluorescence produced from the toxin, and screen for toxin production using that. Well, when we tried that, John said just mix it 1-to-1 with water, put it in the plate, put it in your fungus, and you'll get fluorescence if they make, no fluorescence if they don't. Well, what we did is took a bunch of random isolates that we got from a consultant, Harold Lambert. He had a client who had applied Aflagard to a field, took the corn to the elevator and was rejected for aflatoxin. And he came to us wanting to know, could we tell him whether or not Aflagard was in any of the kernels? To make a long story short, we couldn't, but these are some of the isolates that we got from that effort. So what we have here is a 24-well plate. We put a 1/2 ml of this medium in there, put them in the dark for 5 days at 30 degrees, and then we sampled the wells for how much aflatoxin was there. Notice the yellow ones have aflatoxin. This one, this one, this one, this one, and this one. These white ones don't look like they have much yellow, and they don't have any aflatoxin that we can detect by HPLC. So we think this is a qualitative assay. We haven't tried to quantitate this yet, and I'm not sure that we can, but we think it's a good plus-minus assay for does this strain make toxin or not. Just put it on Coco Lopez 1-to-1. This is, by the way, is the bottom of the plate. Well, why is that? What are we looking at there? Turns out one of my colleagues up at Stoneville, Hameda Boss, published a paper some time ago showing the source of this yellow pigment production. What you see here is the aflatoxin gene cluster, the genes that it makes, and the biosynthetic pathway for aflatoxin. And these yellow marked ones here are anthraquinones, which are intermediates in the pathway of aflatoxin synthesis. If you think of this like a Ford assembly line, it makes a Ford and comes off the end down here. And it's the same way you build aflatoxin by adding to the previous substrate another constituent until you get aflatoxin. But apparently what happens here is the Ford on the assembly line gets to here and it goes out the side door, and it gets to here and this unfinished Ford goes out and this unfinished Ford goes out. So you get deposition into the medium of these intermediates which are yellow, and this is the basis of this yellow assay that I'm showing you about. Well, what other use is this? This is our toxigenic 53 was put in all these wells. We put various biocontrols in there, 51, Aflatoxin, 17, 19, AF36. Put them in the dark for 5 days at 30 degrees. No yellow. Intraspecific aflatoxin inhibition due to touch. Okay. What's going on here, I don't have an explanation for. My HPLC was broken at the time, so I can't give you the aflatoxin levels here. Whether my isolate here was not performing well or dead or whatever, I don't know. I need to repeat that. But what it suggests to us is we have an assay now that we can look at toxin production or no toxin production. And we have an assay where we can screen for strains which can be used as putative biocontrol strains. Okay, let's start over. Uses of the yellow assay. Quick qualitative screen for aflatoxin production. Quick qualitative assay for detecting isolates having intraspecific aflatoxin inhibitory ability, touch inhibition impaired. These are putative biocontrol strains. The real use we want to make of this is as a quick assay for atoxigenic mutants impaired in intraspecific aflatoxin inhibition. Normally when you put the toxin and atoxigenic together, you get white. If we mutate something here, put them together and get yellow, that means we've altered something in here that signals this one to shut down. Conversely, if we mutate toxigenic strains, put them in and show that they can make yellow, then pair them with a atox, which normally would turn the well white or leave it white, and we see yellow, that means we've mutated something in the toxigenic strain which is involved in receiving the signal to shut down aflatoxin synthesis. So that's what we hope to begin as a quest to look at the basis of this touch inhibition, which we think is the basis of the mechanism actually of biological control. And pie in the sky by and by, if we can understand what these ligands are that interact to receive the signal and to shut down synthesis or to send the signal, long after I'm gone, perhaps they can put that into corn. The fungus comes in and grows, sees that signal and says, "Oh, I can't do it, I can't make toxin anymore, I'm done." And that may be a possibility. So this is the summary slide. This is what we understand. This is biological control according to Damann, okay? Not everybody may agree with this. I think there's what we call parasitic biocontrol. This occurs in the infection court. In the silks, these spores fly up here, the atoxigenic one lands and begins to grow. And I'd almost call this competitive inclusion, not exclusion. You got to have them both there to get the inhibition. Or you could call it competitive phenotype conversion by touch inhibition. And I think that is really what's going on in here to suppress aflatoxin in the system. What I refer to as saprophytic biocontrol or competitive exclusion is what we've talked about before, is the population distribution in the soil. I think just because you have competitive exclusion in the soil is not necessarily required or sufficient to bring about biocontrol. If the strain that is in there and is competing is one that can fly, can infect, and can inhibit by touch the ambient toxigenic strains, then that's a good thing. This provides an epidemiology basis and sustainability. I think Peter alluded to that a minute ago, the sustainability aspect. So that's all I have for you today. I'd be happy to try to answer any questions.
Questions? Sir?
Kenneth, I do like your touching, but how the touching to answer the question of the replacement we talked about.
Your question is, how does touching, I'm not quite clear on your question, Will. Give it to me again.
So here you have the touching,
Yeah.
but we also talk about replacement. You know, how you compare those, your touching theory and this replacement theory.
Well, well, I can talk to you about this later. The displacement theory, the physical exclusion is what you're referring to, I guess. Is that right? Well, if you looked at Hillary's data, you don't see much physical exclusion. You see growth together.
So which way you go? So there's a touching or replacement. Touching, you think touching is a primary mechanism?
I guess I'm having trouble getting you. We don't know what happens after they touch. We've shown in the first 24 hours of the toxigenic strain's growth, it needs to interact physically with the atoxigenic strain, and not all atoxigenic strains will work.
And what I'm saying here is that based on Peter Cotty of Biocontrol last 10 years, we talk about this replacement. You know, because Peter Cotty has the data many years.
He's got a lot of data for presence in the soil. Isn't that right, Hillary? And some in the kernels. But I think the real mechanism is in the interaction in the infection core. That's what mediates biocontrol.
And sometimes you're very convincing, I agree with you, but there's a plate I say you're 51 and 53, then it doesn't work. That's kind of wondering what's going on.
Well, this gets to the point, again, we have to know what the aggressive infective isolates are in the soil, and we have to know what strains that don't make toxin can fly, infect, and inhibit.
But your theory came from your 51 and 53.
This applies to many other toxigenic combinations. We've shown, for instance, if you look at 3357, AF70S, and 53, that different atoxes inhibit some of those and not others. It's a patchwork, okay? Just because it inhibits A doesn't mean it'll inhibit B. This is more complicated. Nature's a lot more complicated than we simple-minded humans can understand. Yes, Ron?
Do you know anything in the literature where they've looked at isolates in the ears, or where they find more than one isolate, more than one, say, toxin?
Yeah, I think that's common, that you have multiple infection. I think that's been demonstrated. Again, the frequency of that and the SNP work that Peter's lab does is the ideal avenue to get at that answer. I think that would either support or deny what I'm promulgating here.
So, with my
Are you saying
simple
that the lineage, let me ask you a quick question. Are you saying like Peter's work, you got these two different lineages, the alpha-bar lineage and the AF36 lineage. And the concentration from by latitude, I guess. So I'm trying to understand where you want to take this. To be honest with you,
Yeah.
would we be better off trying to ultimately identify the mechanism in which touching inhibition works?
That's
So
our goal.
that we can
that's a possibility, yeah. I'm not gonna do that, but perhaps others will if we can find it, okay? Yeah, David?
So, in this pina colada mix, you're growing aflatoxin in it. Is that what I understood? I mean, you could test that dish and tell what parts per billion of aflatoxin is present.
I don't hear well, and you have
Okay, in
to.
your
Yeah.
pina colada deal,
Yeah,
where you put it, you said these have aflatoxin.
yeah.
Do they have a flavus, or do they have aflatoxin that's measurable?
Well, when you, in each of these, we've introduced the canidia, they grow. If they're aflatoxigenic, they put out those yellow intermediates and they turn it yellow and they make aflatoxin. I've tried to look, for instance, at AF36. Apparently has a lot of the pathway enzymes in it, or genes in it, to try to see whether or not, by some strange reason, if it is turned on and makes those and it just doesn't make the final. I'm not sure where the lesion is off the top of my head in the 36 pathway, but I've looked at a number of these, looked at the NOR isolate, but you can't tell anything about the NOR strains because they're all pink anyway and red, another intermediate that's blocked. But I'm trying to test whether this is true or not. In other words, are there atoxigenics out there in which the pathway is turned on and doesn't get to the end and puts these out that would give us a false positive? But I haven't been able to find that one yet. May be there, may not be there, I don't know. Did I answer your question? Or?
What I'm trying to get at, I thought that in this process, the corn kernel became infected with aflatoxin.
But it became
In theory-
infected with the fungus.
In theory, the aflatoxin is not in the kernel. It's growing on the kernel, feeding on the kernel.
I think, my understanding of conventional wisdom is, and somebody correct me if I'm wrong, that this fungus comes in, colonizes the embryo tissue predominantly, which is rich in lipids, and then can maybe move on and do a little bit in the endosperm, the starchy area. But it has to, in the corn, it infects that kernel. It goes in there, eats that embryo, and pumps out aflatoxin. And that's what gets you rejected at the elevator when it does that. If we could, it did that. Did I answer your question? I'm not sure I understood
Yes,
it.
I'm just trying to figure out if you can grow aflatoxin in a petri dish,
No, we're growing the fungus.
but it's
Fungus
producing
is making.
aflatoxin.
Yeah, right.
So really, this is more of an attachment to a kernel than it is the kernel really being aflatoxin. I mean, then it becomes a cleanup method to get rid of it.
I'm not, it's not
Well,
translated to me.
Yeah. Well, if it's growing here
Yeah,
and it's fed on the garden,
it's
so
got
if you
to
get
eat.
rid of this, you've got rid, the kernel's not gonna really test aflatoxin so much. It's something you can wipe off.
If you could, well, if people could run it through a magic magnet and pull out those that had aflatoxin in them, they tried to do that, but
Is the mycelium inside the kernel?
yeah,
So it's the
Yes.
mycelium inside the kernel.
yeah.
So is Fusarium for fumonisin, does it act the same way on the kernel?
I'm going to have to defer to the expert on fumonisin over here, Bert. Yeah, sure, I'm assuming it is in the kernel.
In the case of, yes, I think conventional wisdom, Fusarium and Aspergillus are a little different. Aspergillus really likes to get to the interface between the germ and the endosperm, and as that sugar is coming across, it almost forms like a biofilm. And then it can eventually rot the entire kernel. But that aflatoxin is getting pumped out. Fumonisin maybe accumulates a bit more throughout the kernel. Fusarium really rots kernel stays in the top. But if conditions are right, it'll really rock that during this room long. Then Mosson's pretty around.
Well, have I confused you all enough now?
Let's stay that way.